This week we're going to talk about the respiratory system. This is a list of the learning objectives that we're going to cover. And we're going to start with an overview of cellular respiration. When we talk about the term respiration, we're referring to an exchange of gases. We all know that we breathe in oxygen and breathe out carbon dioxide. Oxygen comes in, carbon dioxide leaves as a waste product. But what is the oxygen used for in our body? Why do we need to bring oxygen in? Why do we die if we don't have oxygen coming in? So that's what we're going to focus on in the cellular respiration part. So we'll start by talking about what oxygen is even used for in the first place, and then we'll go through the respiratory systems so that you understand how important all of these different structures in the respiratory system really are. So recall that energy is adenosine triphosphate. ATP. In order to make energy, we need to figure out how to regenerate these molecules when they are separated. So when we use ATP, ATP breaks down into ADP plus a free phosphate. We want to regenerate ATP so that we can keep this cycle going. So we break it apart, we remake it, we break it apart, remake it. And ATP allows us to do work in the cell or to create a movement inside of the cell. And we talked a little bit about that in energy way back in the beginning of the semester. So how do we make energy? Well, recall that a cell has a cell membrane. And then in the inside of the cell is the nucleus. And then there's specialized organelles, one of which is referred to as a mitochondria. And we said that mitochondria are the energy centers of the cell. So we're going to learn about the process of cellular respiration and why we consider them the energy centers of the cell. Energy production most easily begins with a molecule called glucose. Glucose is in the shape of a hexagon. So it has 6 carbons, 12 hydrogens, and 6 oxygens. So if we draw the structure of glucose, looks something like this. So here you can see now the carbons, 1, 2, 3, 4, 5, 6. And then you can count the oxygens, 1, 2, 3, 4, 5, 6. And the hydrogens, you can count there's 12 hydrogens. So that's the molecular structure of glucose. I'm taking the time to draw out the molecular structure of glucose so that you can understand what's happening to this molecule when it goes through the process of cellular respiration. Now in this class, we're just going to do a basic overview of cellular respiration. But if you're going on to take advanced A&P, you need to have this foundational knowledge in order to understand every single enzymatic reaction that happens in cellular respiration and its relevance to human health. So, cellular respiration has 3 major steps. The first step is called glycolysis. And that happens in the cytosol. If you look at the term glycolysis, lysis means to break apart. Glyco is referring to glucose. So in the process of glycolysis, Glucose comes into the cell, and then it's ripped in half into 2 molecules of pyruvate. So in glycolysis, glucose enters the cell, and eventually, after 10 enzymatic reactions, we end up with 2 molecules of pyruvate. But there are some important things that have to happen to glucose as soon as it comes in the cell. First of all, glucose will travel down its concentration gradient so that there's an equal concentration of glucose inside and outside of the cell. In order to prevent that from happening and to trap the glucose inside of the cell and force more glucose to come into the cell, we add a phosphate to glucose. So glucose becomes glucose 6-phosphate. Insulin aids in the initial phosphorylation of glucose. When we phosphorylate glucose 6-phosphate, all we're doing is adding a phosphate group in this position. And sorry, I drew it in the wrong position. Let me draw it in the right one. So there it goes, the phosphate in the 6th position of the carbon. Glucose 6-phosphate is then converted to fructose 6-phosphate, to take on more of a pentagon shape instead of a hexagon, and then it becomes fructose 1,6-biphosphate. So this molecule here takes on a different type of shape. And I'm just going to draw kind of the basic skeleton. Now our 6th position carbon already had a phosphate group on it, but when we convert it to fructose 1,6-biphosphate, we add another phosphate to it. This gives us kind of an even stage to rip this molecule in half. So after this step, there's a series of other steps that happen, but eventually it ends up with the production of 2-pyruvate, or pyruvic acids. Now through this process, what we've done is we have burned up 2 phosphates. So we had 2 ATP available, and we used 2 of them in order to produce this. So those 2 that we had were used up. But overall, we ended up gaining 4. So we say that this is a net gain of plus 2 ATP. So we made 4 through all of these different steps, but we used up 2 of them, so now we have the 2 left. And that's the basics of the first step of cellular respiration, glycolysis. So to recap, what's happening initially is all we did was bring glucose inside of the cell. We trapped it in the cell by adding a phosphate to it. We added another phosphate to it so that we could rip it in half. And then once it was ripped in half, that's when we ended with our 2 molecules of pyruvate. All in all, we ended with 2 ATP, so a net gain of 2 ATP. Now those pyruvate are carried to the next step, and the next step is called the Krebs cycle. And there's a lot of different names for this: the Krebs cycle, the TCA cycle, the citric acid cycle. And all of those terms are appropriate. I prefer to use the Krebs term or Krebs cycle because that's what I was taught, but you can call it any one of those things. Exam questions will usually refer to it as the Krebs cycle. So when we begin this happens in the center of the mitochondria, and that's known as the mitochondrial matrix. So these pyruvate that were generated here in the cytosol are now going to be shuttled to the center of a mitochondria. When the pyruvate get in there, they are going to be converted by pyruvate dehydrogenase, this large 3-complex enzyme, to acetyl, which is going to be carried by a coenzyme A. So we call this combined acetyl-CoA. Acetyl-CoA is going to enter into this Krebs cycle. So what happens is oxaloacetate combines with the acetyl, and we start along this pathway of the Krebs cycle. There are 8 separate enzymatic steps that happen. At each point, it's taking this pyruvate molecule and breaking it down into smaller and smaller pieces. When we break this apart, if we want to go back to the original structure, the whole reason why I drew out this structure was to point out that this is made up of carbon, oxygen, and hydrogen. In the Krebs cycle, carbon dioxide is lost as a waste product here and here. Hydrogen ions and electrons are given off, and we end up with a net gain of 2 ATP. Now these electrons are transported to the next step along the way through 2 electron transporters, NAD, which carries 1 hydrogen, and FAD, which carries 2 hydrogen. And these hydrogen protons are left behind in order to build up a gradient. So, to recap, glucose was brought into the cell, glucose got ripped in half to pyruvate. Pyruvate got shoved inside of the mitochondria, and inside of the mitochondria, that pyruvate got broken down into its building blocks. That released carbon dioxide as a waste product, hydrogen ions and electrons, and we ended up with a net gain of 2 ATP. This is where our major waste product comes from, and that's why we have to breathe out. That's why we have to have a respiratory system to eliminate that carbon dioxide. So the last step along the way is called the electron transport chain. And this is going to happen in between the 2 mitochondrial membranes. So you've got these 2 membranes, and then you have this huge complex called ATPase. The hydrogen ions are going to be delivered, the electrons are going to be delivered, and they're going to work their way through 4 membrane complexes. So these electrons are going to kind of bounce through these different electron complexes, eventually coming back into the center here. The hydrogen ions that we're accumulating are going to be pumped into this inner membrane space. So now that we build up a gradient of hydrogen ions in between these 2 membranes, if you remember from our transport definitions, we never want to have a concentrated substance on one side of a membrane. We always want to have an equal concentration on both sides of the membrane. Plus, these are all positively charged particles. They hate each other. They repel each other when they're the like charge. So there's a couple different forces that are at bay here, or at work here, which is why we call this the proton motive force. Because these are concentrated, they want to go back through the membrane. And the only place that they can do that is through this ATPase. As they go through, it causes this thing to rotate. And the rotation of this complex is going to take ADP, add a phosphate to create ATP. So now our hydrogen ions are on the center here, and our electrons have gone through the electron transport chain. Well, we take these electrons and these hydrogen ions and combine them with oxygen to neutralize them and produce water. This is the reason why we have to breathe in. Let's say that we didn't take a breath. Let's say that we didn't have oxygen coming in. What would happen is eventually the inside here would be filled up with these hydrogen ions. And hydrogen ions create for an acidic environment. So now you've got an acid here, and anytime that you have acid, you have enzymatic degradation or denaturation. Enzymes can only function within a particular pH range. So if you decrease the pH, increase the hydrogen ion concentration, you're going to start shutting off your enzymes. This whole system is going to back up, and the Krebs cycle is going to shut down. And we're going to go all the way back to the pyruvate. So now pyruvate will be forced to turn into, in humans, lactic acid. If our pyruvate turns into lactic acid, that's called lactic acid fermentation. And that's humans' type of anaerobic respiration. So this whole process that we just went through, all the way through to the end, is known as aerobic respiration, because we have oxygen here as our terminal electron acceptor. In anaerobic respiration, we don't have enough oxygen in order to accept all of the electrons. So our Krebs cycle shuts down, all of those enzymes denature in the Krebs cycle, and we end up converting our pyruvate to lactic acid. That can sustain our life for just a short period of time. We can produce 2 ATP when we go through this cycle. But if we don't take a breath, eventually our enzymes aren't going to be able to kind of regenerate, and we're going to die. So we need to keep oxygen coming in order to serve as this terminal electron acceptor and keeping this whole chain moving forward. As soon as oxygen ceases to come in, this whole thing shuts down. We end up producing just a small amount, a fraction of the energy that we would need, and we could end up dying from that. All in all, the electron transport chain gives us 32 to 34 ATP. If you add that to the 2 plus the Krebs cycle from the Krebs cycle, plus the 2 from glycolysis, you got 36 to 38 ATP per glucose. And that's for aerobic respiration. But if you go through anaerobic respiration, you only get 2 ATP. Please take some time to watch the videos that I've posted about cellular respiration. I know sometimes it's hard to follow along with my diagrams, but my diagrams are abbreviated, so they only give you the bare essentials of what you need to know about these processes. Questions on the exam are going to be like, your options are glycolysis, Krebs cycle, electron transport chain, and it'll say something like, during which of the steps is glucose 6-phosphate involved? During which of the steps is carbon dioxide released as a waste product? Which of the steps produces the most ATP? And so forth. So make sure you're familiar with what's going on in these different steps.